I'd like to introduce Stephen Gardner. He's been working with me for about a year and a half now, just about a year and a half, semester, 2 semesters. Came to me about August, somewhere in that range, a little bit before that, and was interested in doing some undergraduate research. And with Dr. Perrigan was doing some work with phthalates, and we said, yeah, it'd be interesting testing some of the toxicity with that. So we kind of started some work with that, and Stephen thought that was really interesting. So he's going to tell us about the toxicity of the Dibutly Phthalates to developing Xenopus.
Thank you all for coming. Let's go ahead and get started. So what is Dibutly Phthalate? Well, Dibutyl was a commonly used plasticizer. It was used in adhesive additives, printing inks, or organic solvents, things of that nature. What's a plasticizer? Well, a plasticizer is a compound that goes into a plastic polymer like PVC or something, and what it does is it's not actually attached to the plastic. It just kind of gives it its properties as far as being able to be, it makes it softer, less brittle, things like that. But the problem is it was added to the California Proposition 65 list of suspected teratogens in 2006. A teratogen is a compound that can cause birth defects and malformations and things like that, so it's a bad compound. So why is any of this important? Well, like I said before, it's not actually joined to the plastic molecule, so it can actually leach off, and it's known to collect in soil, sediment, and it's actually a volatile compound, meaning it can evaporate and get into the air. And right now, new research to develop less toxic phthalate compounds is currently being performed. But in order for us to be able to detect how toxic these new compounds could be, we need to have a good means of assessing the toxicity and teratogenicity of the compound for the new and old phthalates. So a possible organism we could use for this would be the African clawed frog, Xenopus Laevis. And the reason why we maybe use this frog is because it's very similar in its genetic makeup and evolutionary history to humans. So any effect that you would cause in the African clawed frog, you could very probably see the same effect being caused to us from this compound. And here's a picture of a Xenopus Laevis South African clawed frog that we have down in the biology department. They're aquatic frogs, if you'll notice the claws on its hind feet and everything. So the way that we would test this is the FETAX test, which is an acronym that stands for Frog Embryo Teratogenesis Assay-Xenopus. And what it is it's a 96-hour toxicity and teratogenicity test that exposes developing embryos of Xenopus Laevis to increasing concentrations of this chemical that we're testing. So the objectives here would be to validate the use of these frog embryos for quick assessment of toxicity and teratogenicity to the Dibutyl phthalate and pre-existing phthalates to these new phthalates that are being developed. Here's our methods. We have to determine concentrations, breed the Xenopus, then once we have them bred, sort their eggs, expose them to concentrations, then record data off of that and interpret what that data would mean. The concentrations, we use 1 to 20 ppm, which stands for if you had 1 penny out of let's see, milligrams per liter, so that'd be 1 per million, so 1 penny out of a million pennies would be a good way to look at that. But the thing is, it has a very low solubility in water, so we had to use DMSO to make sure that it stayed in solution and that we could expose at the concentrations we wanted to expose at. So DMSO stands for DiMethylSulfoxide, and what it is it's both a polar and non-polar compound. What it is water is a polar compound, so things that are not polar don't react very well with it, so it would just kind of sit at the bottom of the container we were using. So the reason why we use DMSO is because it interacts with the water molecule, but at the same time, the non-polar methyl groups on it will interact very strongly with the Dibutly Phthalate that you see at the bottom left-hand corner. Another reason why, so it keeps it from falling out of solution, and another reason why is because it very easily crosses cellular membranes. So anything that's in solution is going to carry with it into the cellular membrane, so that you definitely are knowing you're exposing these developing embryos to this compound. So we bred the Xenopus on Sunday night and injected them with this hormone to induce them to breed, and they were allowed to breed over the night. And this is the breeding chamber that we put them in, and as they lay the eggs, they fall through the scrape and collect in the bottom of the container. So then Monday morning we'll come in, we'll get these eggs, and we have to get rid of the jelly coating that surrounds them because that can interfere with compounds getting into them, and we don't want that there. So what we do is we use the amino acid cysteine, we get it to a pH of 8, and it will go in and actually dissolve the jelly coat. So once we've done that, we have to sort the eggs, and what you want to do is you want to have properly developing fertilized eggs, because the whole goal of this test is to prove that this chemical is causing bad effects, whereas if you tested on a bad egg that was already about to die or was not developing properly, it doesn't really tell you a whole lot when it gets malformed or something. So then once we have our eggs, we place 20 viable eggs into each dish that we're going to run. The picture on the left would be a cluster of eggs, and the big white ones there are either not fertilized or they're developing improperly, so we wouldn't use those. And then on the right, we see the ones that we would use for this test. So we ran 2 sets of controls, a FETAX only control. FETAX control is just the solution with the ions in it that these guys really need to survive and live like they should. And then we ran a DMSO control with our highest amount of DMSO, because DMSO is known to by itself at a certain concentration to cause malformations and to cause problems. So we wanted to be sure that our DMSO we were using to keep this in solution wasn't causing anything that we observed in the test. Each day I would come in and make new solutions and switch them out with the existing solutions that these eggs were in, because if you leave them in an existing solution, it gets kind of dirty with them, you know, with their waste that they put out and stuff like that. And also if there was anything dead in the dish, it releases chemicals which can kill other tadpoles as well. So each day I came in and removed mortalities, made a check mark, you know, I counted the mortalities, and then recorded the pH, because if the pH is too high or low, it can cause problems as well. So at 96 hours that Friday, pH and malformations are recorded, and in order for me to record the malformations, at this point they're swimming around in the dish. So you have to kind of anesthetize them or knock them out so they can you can get a good look at them under the microscope. So we use a compound called the MS-222, and once they're knocked out, we take pictures of any abnormalities that are present, we record the abnormalities that are present, and then we take pictures for length measurements, and we run that through a program called Image Pro Plus, and it records how the size of each tadpole, because we want to see if this compound will stunt their growth at all. So then once we have all this data, we have to interpret what it means. So all this data gets sent into a program called Systat that will analyze every single bit of the control, every single bit of the concentrations we tested with the controls to tell us how statistically different they are. And then from that, we obtain the LC50, EC50, and no observable effects data. And what LC50 stands for is the concentration required to kill 50% of the population of our tadpoles. So if you had a dish with 100 tadpoles in it, what concentration would you have to hit those guys with to kill 50 of them? The EC50 stands for the effective concentration required to malform 50% of them. So same scenario, if you had a dish with 100 tadpoles, what would you have to hit them with, what concentration to make sure half of them get malformed? And then the no observable effects data, at what concentrations can you have that no effect will be observed from exposing it to? So the results were, we took our DMSO and our FETAX controls and compared those two, and there weren't any real statistical differences between FETAX and DMSO, which you would want to see, because that way we know that the DMSO we used to keep this stuff in solution and as a carrier solvent didn't cause any of the mortalities or malformations that we saw later on in the concentrations that we tested, so that's good. This is a curve that we generated. If you look at it, the black dots, as they go up, that's the curve for the mortality. So as you see, as the concentrations go up and increase, the mortalities increase until it hits the very top of the graph, which is 100% mortality. Then you have the second slope, which is the malformation curve, which it steadily increases as well, but there's that little area between the two. And the area that is between the two curves, slopes, would be the teratogenic index. The teratogenic index stands for the difference between the LC50 and the EC50 slope, and that tells you how much more likely you are to malform something as opposed to kill something. So if something like cyanide, for example, it would have a very, very, very small teratogenic index, because if you get hit with a little bit of that, you're dead. You're not going to get malformed from it, as opposed to this compound, which we found out and later on, it's more likely to malform you than it is to kill you. And then as we get, the way we get our LC50s and EC50s, if you see the line coming across it at the 0.5 mark and then going down right at the first one, that would stand for our EC50 because it's taken off the linear regression slope you see there for the malformations. And if you follow it all the way to the end, you get the LC50, which is determined from the linear regression slope for the mortality curve. So then here's the growth curve. As I mentioned, we take pictures of the lengths of these guys. So as you see, as the concentration of DiButyl increases, their lengths decrease. So this compound does, in fact, stunt their growth a little bit. Here's the results again with the LC, EC50, and teratogenic index. There's 95% confidence intervals for each one of them. That just says that we're 95% sure that this value lies between this range. Like if you take the LC50 for the first one, we're 95% sure that the LC50 is between 10.464 and 12.676, and that's the way that works for all of those. So here's some pictures of controls. This is what a standard tadpole for Xenopus Laevis would look like at Friday at 96 hours. You can see kind of the gut coil, the gut, that yellowish part at the back of the tadpole, that would be the gut. And you see the head and thigh and everything else. Well, here are some that were malformed at a higher concentration of this chemical. Those that I just showed you a minute ago were the controls. This is at a high concentration of this Dibutly Phthalate. If you look at it, you see some edema at the bottom portion of the tadpole, and you also notice that the gut is completely coiled differently than in the controls here. So its gut is very distorted. It's got a lot of edema, and also it's got that red is blistering on its tail. And the other one on the right, it also has the same edema, and it has the gut coiling malformation, and also it has a notochord, or the spinal area. It's kind of bent upward at a little bit of an angle, so that was another malformation we observed. So conclusions. A teratogenic index lower than 1.5 would indicate that it's non-teratogenic. Well, if you take all of the TIs we have here and average them together, we wind up getting a teratogenic index of 1.56, as well as having the malformations from the pictures that I just showed you. So we can conclude from this two things. Number one, that these frog embryos can be useful in assessing the toxicity of these phthalate compounds. And two, that DiButyl phthalate is in fact a teratogenic compound. So possibilities for future work would be determining the critical time for exposure of this compound dissolved in DMSO to Xenopus Laevis embryos. And what that would mean is, instead of exposing them all Monday through Friday, just expose a dish, like let's say on Monday, and then leave it alone the rest of the week to see if it had any of the malformations, or expose it on Tuesday and see, so kind of what time these malformations are occurring with these embryos. Another possibility would be how it's interacting with the Xenopus Laevis to cause these malformations, and again comparing the effects of the different phthalates based on their size of their molecular size. So I'd like to thank Dr. Rayburn and Dr. Perigin for letting me work in their labs and for kind of getting me into this research project and teaching me what to do and things. And are there any